Wow, welcome, everyone.
Hello.
Hello.
Thanks, Rich Turfry, also known as Buck 65. Zombie Delights is now my go-to Halloween tune. That's pretty awesome.
It's not the Monster Mash.
You do the Monster Mash. No.
Welcome.
And don't even say those titles because that's
Earworm?
unfriendly earworming.
Yeah, sorry. Okay. Hi. We
Hello.
hope you're okay. Have fun tonight.
Yeah.
Because we all deserve to have a little bit of fun. Be safe. Wear a mask. Speaking of masks, please wear them. If you need some evidence, we have evidence. Please wear masks. Yep. And make them scary tonight.
I mean, if there was a zombie apocalypse, you'd protect yourself, right? You get the cricket bat, or you'd get the hammer, or you'd go find like the oddly preened people of The Walking Dead with their crossbows
and their plucked eyebrows,
and their tomatoes living for decades on farms that make singularly tomatoes. You would do all those things, right? And now, never before in the history of human epidemics have so many been asked to do so little. And tonight it's a freaking holiday about wearing a mask.
So wear a mask. Under your mask. Enjoy. Have a good time.
It's a double mask.
Okay, I think we have to acknowledge the 8. Because pumpkin number 8. Because I think we're there too. It's hard. It's
I identified it. I self-identified as an 8. Yeah,
we're there too. Everyone's there. All of this is really hard, and so we totally see you and we feel you. And there are things you can do, like have a good time tonight, and little bits of kindness that you can give to yourself in order to help get through it. But it's not easy. And it's funny, they say it gets better. It's kind of a marathon. It doesn't necessarily get better soon. What I can tell you though is if you are in first year, most of you are, not all of you, but if you are in first year, it's not that it necessarily gets better. It's that what's happening now matters less the more you move forward. So you learn how to cope with these things. You learn how to, run a marathon, not a sprint. And ultimately these things, though they feel like they are super important, and they totally are, but what I'm saying is that importance will kind of like diminish over time. I published my first year university grades not too long ago on Twitter. You'll see. It's okay. I'm still employed. I have food. I have a great family, a house. Everything's fine. Even though my biochemistry grade was not amazing because, oh my God, I hated biochemistry. Okay. That's all I'll say. I just really don't want to delete any of these drawings because they're lovely.
Shout out, yeah.
Yeah. That pumpkin at the top is pretty creepy. Thank you, Noor. And Sam, this is amazing.
I'm not sure it's a pumpkin. I think it's something that's leaning in from the darkness. This is the abyss looking back at you. Cue your Werner Herzog's "This is when you look into the abyss."
Okay.
The abyss looks back.
Great.
I have to practice my Werner.
Okay.
That's better. Werner.
Let's get on to the biology because some of you are going, guys, stop chattering. Just get to it. We will get to it.
Zombie delight.
Okay.
Zombie delight.
Let me just find. There we go. And we're gonna clear. Okay. So today and on Wednesday, we're going to kind of wrap up all of the ecology stuff because we have a topic test coming up. About the topic test, you will not be surprised by anything. The idea is that basically the first one was a practice test, although if you did really well, you get to keep the grades. If you totally blew it, no worries, we drop that grade. So this one coming up, is about ecology. So it's just going to be on the ecology stuff. But now you know how we write tests, right? So now you know how you need to study, or hopefully a little bit better. We've posted the asynchronous study tool. The link is in the announcements. You can get started there. You'll notice that there are no prompts. It's because you're meant to go and put in the prompts now. So the best way to do that is to take a look at all of the vocabulary that we've talked about. Competitive exclusion, carrying capacity, inflection points, all these things, and like see if you can write a true or false statement about that artifact. There are way more sort of sheets for you to play with and to practice on, and really try to get some kind of conversation going with other folks.
And remember, I see that there's a Quizlet that you're making for this, which is great. That's a great way to get together in groups, kind of virtually broaden the specific group that you're getting together with. And remember, your experience in that first topic test was that we did not quiz you on the facts, right?
Yeah.
We will give you some facts, and then we will ask you about them in a way that is appropriate according to the kind of learning objectives of that unit, in this case, the ecology one. So move beyond the memorizing and into the synthesizing.
Yeah, and we're going to do that today when we take up the homework. We're going to take like an extra long time on the homework, and we're going to walk you through a bit of an exercise that I hope is going to be helpful.
Our Tail 20, is there an option to write the topic test on Friday?
Yes, you can write it on Thursday or Friday all day from 9 until 6 p.m. All of the details will be posted on course link. So yeah, your Thursdays really suck. So we're going to do a Thursday, Friday option again, but this time it'll be the full Friday because it's not Thanksgiving. So yeah, just make sure though, if you want your 3 hours, you sign in at 3 o'clock or before so that you can have until 6 p.m. We will close it on Thursday night. Because we're going to be sleeping, and so, you know.
As soon as possible.
Yeah. Anyway, all of the information will be available or already is available on the announcements. Okay, good. So let's talk about the homework. So this was your homework.
It's homework from several days ago. This isn't Wednesday. We kind of stockpiled some. So these are your Monday questions.
Yeah.
And we'll get to your Wednesday questions on Wednesday.
Yeah. Okay. So we did some of this stuff already, just kind of following the line and whatever. But now we want to kind of bring it all together. Carrying capacity, independent and dependent treatment options, or population independent or dependent treatment options. Okay. So what we did was we kind of designed a bit of an assignment to walk you through some of these things, and then hopefully there was an aha moment, but if there isn't, you're going to have it today.
Take on me. Take on me. That's my aha moment.
That's your aha moment? Okay. Okay, so what we want you to do is, let's use hold on just 1 second. There we go. Okay, using a red line, please go ahead and draw for us a density-dependent control treatment.
Questions about format.
Great. Super. Really, really good. Okay, now, for those who are ready, let's switch over to a blue line and do an effective density independent control treatment. And then those who are ready for a green line, an ineffective density independent control treatment. Very good.
I love it.
Very, 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 very good. Okay. Super. The vast majority of you are getting it. Super good. So the idea is, and you know, you can be a little wavy or curvy, that's totally fine. For the density dependent control treatment, the idea is that it does depend on the density, which is on the x-axis. So that's going to be the only one that's going to change with the x-axis. Everything else is not going to change with the x-axis. So either it's effective or it's not. So it's going to be high or low. Super good. Obviously, when the population gets to like 0, it's not going to kill anything, but it's still going to be effective, right? And so that's the thing that we want you to decouple, is the mechanism from the outcome, okay? And that really just requires you to take a look critically at what is on those axes, right? So if we were to look at not population density, but total population size, that would be a little bit different. If we had it instead, the treatment was on the x-axis and the population was on the y-axis, again, those would mean different things, right? Your independent and your dependent variables. All of these things really matter when we talk about, you know, effects and making predictions about what those patterns are going to look like. So you've done excellently. I am going to clear it, annotate clear, and we'll go and simplify it a little bit. So if you have any questions about that, please pop them in the chat, and we can answer them as we go. But let's imagine then that this is the pattern that we're looking for, right? Where with density dependent effects, it relies on, or it changes with the population density on the x-axis. So given that, we have a polling question for you. So we've brought that down here, and here you go. Just hold on. Take a look, read through. I'll launch the poll for you. There you go. How are we doing? 65%. Give you just another few more seconds.
Some of you are practicing the element of the topic tests, which is to read the question a couple of times and to determine whether we're asking for the thing that agrees with this or the thing that doesn't agree with this. And of course, you're doing it in a low risk environment right now because we're not recording this. You know that you read it not in the way we wrote it, and that's fine. And so use it as a way to jot in your notes. It's like, I wrote that. That's clearly one of the blah, 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 not the blah, 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 which is what I wanted to write.
Blah, blah, blah.
Blah, blah, blah.
Okay, good. Okay, so we're reaching a bit of a saturation point with contributions
80%.
here.
80%.
So we're going to share the results.
Boo.
Okay, good. Hold on to this, okay? So we're not going to give you the answer right yet. I think we're going to give you another chance to fill this out. So just hold on to it. Most of you are saying the thing that is not consistent is treatment A is the best option to eradicate the population. So what you're saying is treatment A is not the best option to eradicate the population. But let's, given that there's quite a bit of a Gaussian distribution, let's.
It's almost an Evenian.
Yeah, an Evenian. Let's take a look and do a little bit of kind of studying together, if that's okay. So you're going to need to.
Wait, you've got a thinking cap.
I have a thinking cap.
Yeah, where's mine?
I think I can.
Right.
Okay. No, not this. Let's go back to a blank. Okay. Okay, here's what I want to do. I would like to change these axes a little bit, if that's okay. So I need to get some annotation tools. There we go. Annotate. And I'm going to can somebody cross population density out for me? And I'm going to put in a new one. Thank you. And then can somebody cross out effectiveness of the treatment? That's great. Thank you. Okay, so we're going to do population abundance. And here, oh no, shoot. Population abundance is going to be here. And down here, thank you. Yeah, cross out population abundance. Thank you. And then we're going to put time here on the x-axis. Perfect. This is what I want to do. Okay, thank you. Good teamwork. Okay. Okay, so now we're changing up the axes, right? So we have to like change our frame of mind. And I'm going to tell you a story. And what I want to do, what I want to happen as I'm telling the story is for you to graph the story. So it's going to be a time. We're going to start at time 0. And then we're going to, go through the story. And what I want you to do is over time, graph the population abundance. Okay. And you can use little dots or whatever. It's not a problem, and it's going to get messy, and that's going to be delightful. Okay. What should we use as the story? Emerald ash borer?
Sure.
Okay, emerald ash borer, because I think that stuff is coming up in the online content stuff, right? Okay, so we're going to talk about the invasive species of emerald ash borer. Okay, so it's killing ash trees. It's a big problem in the world. And so obviously, if it's an invasive species, at time 0, there weren't any, right? So let's, thank you. Good. Okay. And then, of course, they came over in crates and all sorts of things from various different places. But obviously, the population that was coming in wasn't a super lot. And so it was kind of pretty slow and kind of, just a few individuals coming. And then at some point, it reached, a population size that allowed for a real kind of explosion in the population, right? So there was a big increase. Super good. I'm going to need you. Yeah, stick to more time. Okay, good. Okay, excellent. That's really good. Okay. And then at some point, it's going to kind of level off, right? Because it's going to hit carrying capacity. Excellent. Okay. Perfect. So that is your very traditional S curve. And what I'd like to do actually is I'm going to delete and I'm going to just kind of slide it over because I need more time on the x-axis. So here we go. I'm going to clear all of these. And now I'm going to draw, if that's okay. Let's do basically a bit of a smush. Okay. So there you go. There's our S curve and it's smushed. Okay, cool. Now we're at carrying capacity. Okay. And so, obviously the emerald ash borer really sucks and we're going to try to, to get rid of it or to manage it in some way. And so this is what I want to do. I want to explore the effects of a density dependent or density independent. Oh, thank you so much, Shannon. That's great. Density dependent or density independent control factor and what that looks like with respect to things like carrying capacity. Okay. So Cool. So the first thing that we're going to do is we're going to try a density dependent treatment factor. Okay. So let's imagine that we find a tree, an ash tree, and we try to infect one of the individuals with some kind of disease that, you know, kind of kills when they come into close contact with each other. So what's going to happen to the population? So it's an effective one. What's going to happen to the population when we first introduce that thing? So we're going to start out at carrying capacity and we're going to go down. Good, Isabella, good. Yeah. Okay. But then remember, it's a density dependent treatment factor. So once we get to, say, this like critical point, what's going to happen to the effectiveness of this disease that we've introduced into the emerald ash borer? Yeah. And then the population is going to rise a little bit because the treatment isn't very effective. So then what's going to happen? Yeah. And then the population density is going to get to that point where the treatment then becomes effective. So now what's going to happen? Yeah, good. Okay. You're getting it. That's great. Okay. Cool. So what I want to do now is I want to smush that over. Okay. So the idea is that it fluctuates. Now, don't always look for fluctuation, right? But the overall effect. Okay, so here we go. I'm going to clear, and what I want you to do, I'm going to put back the S curve, right? Okay, and what I want you to do now is to kind of summarize a bit. So we've got the S curve, we've got the density dependent, so it's going to go down. And what's it going to do? Hold on, for some reason I don't have my pen. It's going to come like this, and then say we introduce the density dependent. Sam, really good. And can somebody summarize Sam's line to kind of like give us just like a trend line of what's going to happen? Yeah, Sam. Good. Good, Danielle. Good. Good, good, good. Yeah, exactly. Winston, great. Okay, so it's going to come down like this, right? Now, within here, there might be fluctuations for sure, right? But we may not see those depending on how frequently we sample or whatever, right? But what can we say in words is the overall effect of introducing a density-dependent treatment on carrying capacity? What happens to carrying capacity? Yeah, Michelle. Good, Lauren. Yeah, that's great. Okay. It decreases. So this was the original carrying capacity. Here's our density dependent, right? And whether it's wiggly or straight, it doesn't matter. All we're looking for is the overall decrease in carrying capacity. So can you use a density dependent treatment factor to eradicate a species? Really good. Nope. Yeah. Do you see why? Because once you get down lower in density, the treatment doesn't work anymore. Okay? That's the theory. And I hope you're thinking, what about, you know, whatever. That's great. What abouts are great, but that's the theory. Okay. So let's imagine then that instead we introduce a density independent treatment factor that is highly effective. So here we go. Up. Yeah. Up. And then really good. And then super effective treatment factor. We're going to introduce it here. What's going to happen to the population? It's going to go down. And as long as it's like 100% effective, we're going to completely knock out the population. Do you get it? Are you good? Thanks, Shannon. Okay. Yeah. Well done. Anything else you want to add?
Nope. That was great.
Okay. Can we
Thanks
do that?
for relabeling the
exercise. Yeah, that's really great. I hope that was helpful. What I want to do is relaunch the zombie question and see if that's okay. And don't worry if you're like too tired and you're like, oh, we'll go through this later. I think I got it, but like, whatever, don't worry. But let's see how everyone does on the polling question again. Where did it go? There it is. Okay, hold on. I'll launch the poll again. Oh, wait. Relaunch poll. There you go. Good. Really, really good. I'll just give you another few more seconds. So we're looking for the thing that is not consistent. Really good. Okay. End poll. And very well done. Good. Super good. So the answer is treatment A is the best option to eradicate the population because it is not the best option to eradicate the population. So we're looking for the thing that is not consistent here. Okay. So treatment A is the best option to eradicate the population is not consistent because treatment A represents the density dependent population management strategy. Okay, good. If you have any questions, pop them in the chat or stick around after class so we can go through all of that again if you need to. Thank you. And?
So before we leave that, as you're thinking about your study groups for your topic test, that kind of thing, we talked about the Quizlet that you're making, urging you not to just kind of to memorize things. We spoke kind of in a throwaway manner last week about zombie examples and saying, yeah, gather all of your pop culture zombie examples. It's not really a throwaway suggestion. If you do that, you're going to get a whole bunch of different conditions that you can graph like that you can compare and contrast and then ask each other questions about the rate of spread with the World War Z versus the Shaun of the Dead versus the blood quantum, that kind of thing. There's a whole bunch of kind of very easy for you and kind of fun because then you watch the films afterwards or before, ways to figure out scenarios that make you think about the topics that we're talking about that then let you quiz yourselves and your friends. And we know that this is an effective way to study because it's kind of what we do as we're writing the test, right? Please don't lick my face or my hands. So, and now for something completely the same as weeks ago. Biomes. You remember them?
Yeah.
So we talked about biomes, and we talked about them in the context of thinking early in this unit of ecology about the importance of, or the role that abiotic conditions plays in looking at the distribution of species and the conglomeration of species, the aggregation of species into communities across landscapes. And one of the cool things, I think, one of the cool things that we plotted out of this was these kind of global maps of biomes that more or less, when you kind of take off your glasses and you look at this and you're like, well, yeah, that's kind of matches what we see. And then we zoomed in together and we're like, oh, but maybe not at the really local scale or even the regional scale where other things modify or even contradict the things that we might have predicted with abiotic conditions. And that's when we kind of enter zombie land and moose land and Newfoundland and Labrador and all of these things. Now, one of the elements that we spend a lot of time with, so this is then balancing the abiotic and the biotic, right? In the first unit in evolution, implicit to that a whole bunch of times was time. And that's really important here too, because when we look at these patterns across the world and we zoom in and out at physical scales, you have to remember, we must always remember, we have to take like the ecologist's creed of saying, I will always remember that I'm looking at a snapshot and this is a thing over time. And if you dial the time, if you're a time lord in your TARDIS and you dial it kind of way out, sometimes things are not just like, well, it's kind of like a glacial recolonization following the melting of glacial ice, and you're like, okay, well, I kind of get that. Sometimes things are bananas. Like bananas. Where if you look at this, where if we take biomes and we group them together and we wanted to talk about, well, there's the Palearctic and the Nearctic, and then we're going to put that aside for all of our rising bile about the fact that the colonial, the Paleo, what are we talking about there? Old in terms of who? But we're grouping biomes, and we've got a neotropical and an Antarctic and an Ethiopian and an Australasian, and then we've got, wait a second,
Antarctic in New Zealand.
And Antarctic in Chile and Argentina. And there's no Antarctica on the graph. And so you're like, what? WTF or WTA, where's the Antarctica? Or why the Antarctica? So when we think about global patterns like this, we need to think about time. Right? And then the big missing circle because of the big missing continent on the bottom.
Let's skip this.
Yeah?
Yep.
Really? Sure. Okay. Skip to the end. You go.
Okay, so time is an important consideration, right? When we see a pattern, we have to incorporate time into our thought process. Okay, so here's a pattern to the Antarctic, but not because it's really New Zealand. Mm-hmm. Here's a pattern. We have this beetle. Tell us about this beetle, because you know about it, right?
This is a weevil, and as we all know, it's not the lesser of two weevils. It's the greater of many weevils, as particularly if you're on the South Island of Middle Earth. One of the cool things, so this is a weevil called Litopilus, I think it's Litopilus huttoni. It's a little tiny weevil that in New Zealand is of conservation importance, oddly, or associated with dunes, sandy environment. So if you're from Ontario and you're learning what kind of think about like something that looked like Presqu'île or Long Point provincial parks in either Lake Ontario or Lake Erie. And their distribution is really divided. They are not evenly distributed across the nation of New Zealand. And if you zoom in, enhance, you'll see that across the Cook Strait, this non-trivial bit of marine environment, non-trivial bit of not great to be an insect environment, there is one population of that weevil that is way up there on the North Island. So, WTW.
What the weevil.
What the weevil.
So, list for us, please, a whole bunch of plausible hypotheses, plausible explanations, right? So we're doing the whole thing. Which one is not consistent? Give us all of the ones that are consistent with being able to explain this distribution based on the things that we've been learning about in this ecology.
And we're not going to, we're going to compile them.
Yeah.
But
what are some of the
go with
stories?
Consistent, less than, so don't let us kind of judge so you can put them right there in that coincidentally wide open tundra of a space, or you can throw them in the chat if you'd like to type faster.
Hypotheses.
I see some already.
There we go.
They weeble and they wobble and they don't fall down. I've been trying so hard not to say that. Thank you.
Well done.
Well done. The weeble is a non-native species. The islands were once connected but drifted apart. Water levels were lower. They came over on a boat. I like the capital. It's like
boat. So good.
Natural disturbance separated a larger population.
Amazing.
There was once a land bridge.
Very good. Okay. For all of these hypotheses, you're all over this. This is amazing. Now, can we up it a little bit?
There's another one. Human habitat modification.
Brilliant.
Okay, up it. What do you want to do?
I want to say, what would you need to do in order to support any of those hypotheses? So what types of studies could you do to figure out which one of these totally plausible hypotheses this is?
So if you got your mechanism, your hypothesis, you could write it again in the chat or on the screen, like mechanism, your test.
Sure. Or just write a test. We'll figure out which one it is.
Examine the fossil
record. Amazing.
Genetics. Recreate in a controlled environment.
Nice. Look at the geographic history of the area. Amazing. Yeah. Good. Okay.
Geological history of the area. Geological studies. Look for evidence.
Okay.
Look at geological theory. Compare land masses from now and before.
Very good. What else?
You are all weevilologists.
How about the boat hypothesis? How would we test the boat hypothesis?
Boat is how I picture that being typed because it was a capital B.
Right.
So it was like,
the boat hypothesis. How would we test it?
What other species were there? That's kind of it. That's compared to phylogeography. So that's really like a whole field.
Yeah. The boat hypothesis. DNA analysis. Boat. Good. Okay.
So if I'm going to shout for boat, I have to shout DNA analysis.
Yeah. So if the North Island population is the result of a boat from the South Island population, what would you expect the DNA to show?
Oh, see, Guanan has already got it here. If the population from the two islands are close, then maybe they recently spread. Transportation records. What would the variation look like?
Check incoming boats to see if they carry the species.
Yeah.
Genetically close, but variation would be smaller because it would be a founder effect.
Yep. Swelshel 1, yep.
Very good.
You got it. So you know what?
Like 1070 is past.
Yeah, we're done. Put your hands up. And like massage your head or massage this person you're sitting beside or pat yourself on the shoulder. Very, very, very good.
Super good. Okay. So can we?
I'm free.
I'm finally free.
We were kidding. You're not free.
Okay. Well, this super good. And
we're still together.
And again, if you're like, man, I can't, like it's Monday morning, like I can't do that. All of the information is there now. We're recording this. You can go through it again. No worries.
So you just came up with a bunch of hypotheses and a bunch of ways to test them. I think of them maybe as sitting under two different categories. In some of the words, some of the lexicon that we've added to you in this unit, they might sit under, yes, the lecture is being recorded. Was the start of the lecture? I think it was, even the ridiculousness with Rich Stir-Fry.
Oh, totally.
See, Rich Stir-Fry. It's totally, when he says his name on radio too, I thought for a long time Buck 65's name was Rich Stir-Fry. It's Rich Stir-Fry.
His last name is Stir-Fry.
Yeah, and not Stir-Fry.
That's awesome. Okay, so let's just summarize these two things into two possibilities with respect to the ecological stuff that we've been doing, okay? So when we say, what could explain this distribution, it's all of those things that can be brought into two big categories, right? One is dispersal, and the other is vicariance.
So your dispersal captures boat because like assisted dispersal. Your vicariance, you've talked a number of times about things drifting.
Yeah.
So splitting somehow. And we actually kind of know some of the answer to this because all of the ways that you just talked about, people have tried to investigate, have gathered data and made conclusions about.
And I love that you brought in that whole idea of genetic variation because that is really one of the excellent ways of doing it. So if it is dispersal, you would expect to see less genetic variation in the North Island population, right? As a result of the founder effect. If it's vicariance, you might expect different genetic variation. If the species was endemic and it's vicariance, they've been evolving over, X amount of time. They may very well be very different that way, right? But you might not see a reduction in genetic variation. You might just see a difference. And you might not see anything, and you might have to do other tests, right? But those are two predictions that you could make.
So you think about that phylogenetically in terms of tree-based thinking. You might see something that was, if it was one of these populations, right? I'm pointing at the screen. Smith, come on, it's been 3 years. If it was one of these very adjacent populations
right.
that led to that, you would plot that phylogenetically, and there might be some kind of variation, but North and South Island would then be stuck together, linked together, probably in the same terminal branch, in the same. And right. If they were separate for a long time, you'd expect them phylogenetically to be separate for a long time.
And so what's the answer?
Well, so you can see the shape there,
yeah.
where it's like we've crossed out the answer. So the answer is that in the Pliocene, the distribution of water was very different, and the shape of those islands was very different. And so the North Island was a much tinier bit of kind of more of an archipelago, and the South Island was bigger. And so the South Island, this where it sits now, on the opposite side of the barrier, the marine barrier of the Cook Strait, was not the way that it was in the Pliocene, when a lot of these, about 1 million-ish plus or minus years ago, where lots of these incidences of speciation were taking place. And so when you were thinking about your hypotheses, this was actually the one that seems to be supported by the data.
By the data. But all of your hypotheses before were supporting the observation.
Yep.
So these are important distinctions, right? So you have your hypothesis, then you do your research, and then you get an answer. But it doesn't necessarily mean that that's the answer for the next thing that you do, right?
And so it becomes yet another example in this course where context matters, and it depends really comes into play. And in this case, it depends is time, and the things that play a role in today's distributions but aren't visible today. And so we come in with our contemporary bias of this is, we see the way the world it is for us today. One of the biases is, well, many species don't see the world the way that we do. They don't see the world at all. They taste it and they smell it. And the other is that the world is a changing environment, and speciation is a process. And so we often can't just go in and say, that is what we see. That's the distribution. Bam! On to the next question.
Would that it were so simple.
Would that it were so simple.
Complicated.
Coen brothers.
Okay. So
we see this on the, you know this.
It's complicated, right? So through vicariance. So vicariance, change in water, vicariance, continental drift, right? Big processes that are happening. This is what vicariance is. Vicariance is damn cool. And there's still a lot of research to do with respect to, these types of questions. What I really like, though, is if we take all of the continents and we stick them together, they form a larger puzzle. And so this is kind of like a no-duh thing, right? Except the guy that came up with it was shamed and ridiculed pretty much for his whole career.
Yeah.
And I don't think it was until, like the 1960s that people were like, oh, continental drift might be a thing, right? People were pretty pissed about it. There was a whole religious movement that was like, well, God made the world perfectly, so how could it be moving? And so there was that to kind of sort through. And there were all sorts of issues and challenges, and I'm going to cough right now, so hold on just 1 second. I'm getting excited.
I'm verklempt.
I'm verklempt.
Want some tea?
No, yeah, thank you.
Hotter than
it's
yours.
Hotter than mine? Can I just pour it in there? Okay.
Is it the same kind of tea?
I think so. Dirty chai?
Yeah.
Mm.
If you haven't tried it, dirty chai.
It's amazing.
It's tea with coffee. It's like a turducken.
Okay, so if we take all of these continents and we put them together, they make this beautiful puzzle that really suggests all sorts of patterns, right? And we do see those patterns, which is amazing. So we see these sort of distributions of species that have gone extinct in the fossil record. What's also really cool is that looking at which species are where tells us a lot about what was going on. So we see terrestrial species that couldn't swim that are in Antarctica and also India. Really evidence that these used to be connected then, if those species couldn't swim or couldn't sort of disperse, right? It's the anti-dispersal hypothesis that vicariance was a big driver of the distribution of the biodiversity that we see in the fossil record. The other thing that the species tell us, especially the plants, is a lot about the environment and the conditions, the abiotic conditions that were there. So we take a look at the distribution of ferns, or this fossil fern that's in Antarctica, this like green swath that goes across all of these continents, and we see them. And I've been to some of the fossil sites in Antarctica, and I've seen the ferns in the rocks. It's bananas, right? As you're freezing your ass off, and you see these tropical ferns. And really what that tells us is about the abiotic condition. So not only were these continents connected, but Antarctica was a heck of a lot more north than it is today, right? So it speaks to, the way that these continents were connected and the environments, the previous environments. And you see all sorts of sort of magical patterns within the fossil record that really tell us the story of the land over time, over millennia and millions of years. Kind of cool. And that's it. Homework. So this is kind of a big bit of homework. We've given you a table, work through it, do the calculations. We want you to kind of fill in the numbers, do the additions and subtractions. It's kind of a big review of all of that stuff. Then take those data and put it in the graphs, and you'll get some curves. And then from the curves, you'll have this here. So it's really just bringing together all of the carrying capacity and assumptions and things and independent and dependent treatment factors and all of that stuff, so that you can test your understanding of it or use it as a means to figure out what you still need to work on.
So have fun tonight.
Have fun tonight.
Be safe.
Yep.
Come back on Wednesday.
Okay. Bye.